Okay, well here we are inside the lab of Hopkinsville Brewing Company. It is a little bit tighter, a little more compact. You will probably want to open up your slides and have them in another window while we're watching this video, just so you can follow along the slides. I know this laptop is going to be hard to see, that's why you'll probably want to have those slides open. So as we get into the vitality and viability of our yeast cells, we're going to need some tools. First tool we'll need is our microscope. We will need a hemocytometer and its slide cover. We will need a couple of pipettes so we can take small samples. We'll need our yeast sample, and we'll need some distilled water. These tools here will give us everything we need in order to count our yeast cells. Alright, so if you used to look at your hemocytometer and put this underneath the microscope, you have two chambers here with some very fine etched lines. That's what these lines are going to look like. Each of these blocks is divided into 9 smaller areas, as depicted in the red here. So you have 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. Each of those 9 areas are broken down into 16 smaller squares, as you see here with the green square in it. Each of those are then again broken down into 16 more squares. And when we're counting yeast, as you look at your slide, you'll see the yellow block here. We're going to be counting 5 of those corners. So zooming in on this center piece here, these are 5 corners, 5 blocks we're going to count. You have your 4 corners and your center. Each one of these blocks is equivalent to 4 nanoliters. That's important. You'll need to know that for the formula here. Okay, so we know that how small what our hemocytometer can hold. So now we need to know exactly how much yeast we have. In order to do that, we need to calculate and thin out or dilute our yeast cells. So the first thing we're going to do is we're going to take 1 milliliter of our sample here. So I'm just going to take my pipette, and I know you can't see it because it's so small, but alongside this pipette there's graduations on there, and you'll see that there's 1 milliliter, 5 milliliters, 15, 20, all the way up to 30 milliliters. So we've got our 1 milliliter of yeast sample here, and then we're going to take 9 milliliters of distilled water. And we're going to mix that in. And we'll give that a little swirl, and that there is our 1 in 10 dilution. Typically 1 in 10 dilution is not enough dilution. You'll have too many yeast cells. It will just be too hard to count how many cells. So you can divide that again. You'll then take your 1 milliliter of this solution and you'll mix it with 9 milliliters here. And we'll give that a little swirl there, and I know that looks pretty clear, but there are yeast cells in there. And that's going to give us our 1 in 100. So what we're going to do now, we'll take a small sample of that, and it doesn't take much, and we're going to put a little drop on our hemocytometer. We'll take our lens cover and just drop it down on top of it. You put a lens cover on there just so that the yeast in your solution doesn't touch the lens of your microscope. And then we simply look inside, we will dial it in, and we'll focus it. And that's your formula. So if you used to look inside my microscope, you'll see something similar to this. Again, remember, we're counting those 5 red squares, the 4 corners and your center. This represents one of those red squares. In order to count your yeast cells, you'll look through it and you'll see your cells. They'll be inside the square here. A rule of thumb is you only count the cells that are touching the lines on the top or touching the lines on the right. Do not count any yeast cells that are touching the bottom or the left. So if you're using this sample here on your slide deck, you'll see we have 20 yeast cells. You count them out: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, and 20. Again, we're not counting the yeast cells that are touching the bottom or the left-hand side. So then we're going to shift our focus over, and we're going to count the top right, and then we're going to count the center, and then we're going to count the bottom squares as well. So in this example, we have 20 yeast cells in the top left, 25 in the top right, 15 in the center, 30 in the bottom left, and 15 in the bottom right. If you add them all up, it's going to give you 105 yeast cells for your sample. So here's your formula. You're going to take your 105 yeast cells, you're going to multiply that by 5 so you can convert it to 100 nanoliters. Remember, each one of those cells, each one of those blocks was equivalent to 4 nanoliters. Multiplied by 5 is going to give you 100 nanoliters. You're going to take your dilution factor, in our case we used 1 in 100, and you're going to convert all of that into milliliters so we can have the total number of yeast cells per milliliter. So you take a 105, multiply it by 5, multiply it by 100 with dilution factor, multiply it by 10,000, convert it to milliliters. It's going to give us 525 million yeast cells. Catch all that? Alright, so let's try it again. We made our sample, we're looking through our microscope, we counted our 5 blocks. In this sample, we have 20, 16, 16, 17, and 15. You add that all up, 84 yeast cells. We still have the dilution factor of 100. 84 times 5 times 100 times 10,000 is 420 million yeast cells. Some of you all may know or make the assumption that you can take that 10,000 and multiply it by 5 and have 50,000. Those are usually numbers to work with. So you could take your 84, multiply it by 50,000, and then multiply it by a dilution factor. I like to spell it out so I don't confuse my dilution factor of 100 or 10 or possibly 200. So I always just spell it out so I know I'm not making any errors. Okay, so in the previous video I talked there's a difference between vitality and viability. Again, vitality is the ability of your yeast to ferment your wort, and your viability is simply your living cells versus your dead cells. So how do we count them? We saw in a previous slide that we just saw yeast cells, but how do we know which ones are living? In order to do that, we're going to use Methylene Blue. So all you're going to do in this case, you use all the same equipment. You're going to take your one sample, you get 1 milliliter of your sample, mix it in, and then we're going to take our 8 milliliters, not 9, 8 milliliters, and mix it in. And then we're going to take 1 milliliter and we're going to mix that in. And you'll swirl that up, you mix that up, you can put the cover back on it, and you can see that it turns blue. That's your methylene blue. So in this case, you'll get a new hemocytometer or clean this one off. And you'll take your sample, place a drop and then another drop, place your slide cover on top of that, place it back underneath your microscope. We'll focus it in. And when you focus it in, the process all remains the same. The difference is your living yeast cells will take that methylene blue and it'll push it back out of the yeast cell, out of the cell membrane, whereas your dead cells, they're dead, they're not doing anything, and they won't be able to dispel that methylene blue. So as you're looking through the microscope, you're going to see something similar to this. It is going to be blue color when you're looking through it. It's from the dye. Your living cells are going to be opaque in the center, and they're going to have a dark ring around them. That's that blue dye that has dyed the yeast cell membrane. Whereas your dead cells are going to look like blue dots. They're not going to dispel that dye. So using the same example or from before, we have our 20 yeast cells. We've got 16 living and we've got 4 dead. Again, we're not counting that dead yeast cell that's on the bottom line there. 20 cells in total, 16 living, 4 dead. We want to calculate our viability. You're going to take your total cells, reduce that by your dead cells, and divide it by your total cells, multiply it by 100, and that's going to give you your viability percentage. So using the numbers from the previous slide, 20 minus 4 divided by 20 times 100 is going to give you 80% viability. That's probably getting to the bottom range of where you want to use yeast to pitch. Again, your individual brewery will tell you what they use and what they don't use. Anything lower than that, you're probably going to want to just discard it and use some yeast, fresh yeast. Okay, so over time, yeast is a living thing. There are going to be mutations. I am not a microbiologist. I do not thoroughly understand this. I do, however, understand that DNA is what triggers mutations. So your DNA is made up of smaller molecules. Those 4 molecules are your cytosine, adenine, thymine, guanine. Typically, those 4 molecules will pair in a certain way. For this sample, the DNA, the guanine pairs with your cytosine, and your adenine pairs with your thymine. So in a perfect scenario, your thymine, your adenine are paired up. However, when you have a mutation, because of whatever factor, we'll talk about them here in a minute, your thymine may pair with your cytosine. And when that happens, you have a new DNA chain, so essentially you have a new yeast. Too many of these, and you're going to experience that in your beer. Different flavors, different characteristics. There's 5 factors that affect mutations inside the brewery. You have temperature, pH, age, environment, and health. Of those 5, we cannot control age. The other 4 we control using tools in the brewery, whether it be with cooling or heating. We can adjust the pH levels. We can certainly make sure the environment is clean, it's not picking up anything else. And we can monitor and control the health of our yeast. The only one we can't change is age. So as your yeast starts getting older, you may want to just discard it to remove the risk of any kind of mutation. You will see in some older breweries in Europe, they've been using the same yeast for hundreds of years. I'm not familiar with it, I haven't brewed over there. But we like to use our yeast here at the brewery, here at Hopkinsville Brewing Company, about 5 generations. After that, we'll just discard it. My microscope can't get into the DNA molecules, so we don't take that chance. So this was just a brief overview of yeast management from the first video presentation to this video presentation. This one we talked about the viability, we talked about mutations a little bit. There's much more out there. There's a whole lot of reading you can do. You can really dive deep. You can analyze and assess and look at different yeasts. But your brewery is ultimately who's going to tell you what you're going to do and what they don't do, and how they manage your yeast. So just take a look at them. Alright, for your reading assignment, there is a whole section within Blackboard, but just a quick overview. We're looking at chapters 5 and 6. We've got a few pages in each chapter. Take a look at them. You will see some of that material on the test.